Hello everyone and welcome to Clinical Cousins YouTube channel. Today we are going to go over the neuromuscular junction. First, we must know that acetylcholine is made out of acetyl-CoA and choline, an enzyme conveniently called choline acetyltransferase combines these two ingredients to make acetylcholine. Now we are prepared to learn about the neuromuscular junction. The neuromuscular junction is a connection between a nerve cell or motor neuron and the muscle cell. One motor neuron can connect to or innervate a few muscle fibers or many muscle fibers. If it innervates a few muscle fibers, it is likely that those muscle fibers are involved in movements that require a lot of control. For example, this would be like moving our fingers and facial muscles. If our motor neuron is in charge of giving orders to many muscle fibers, it is likely that those muscle fibers are involved in movements that do not require precise coordination. An example of this would be the movement of the large muscles of the thigh. The action potential in the motor neuron will cause a neurotransmitter, acetylcholine, to be released into the synapse, and a muscle contraction will be the likely result. Now we will take a closer look at our cholinergic motor neuron, or acetylcholine releasing cell, as an example. As the action potential is propagated down the motor neuron, it will pass its inward sodium current down the cell until it finally brings voltage-gated calcium channels to threshold. As soon as these voltage-gated calcium channels reach threshold, calcium will rush into the cell, where calcium binds to a vesicle that contains a neurotransmitter inside. Our vesicle envelope is then ripped apart as it performs exocytosis, and our acetylcholine neurotransmitter can now diffuse across to the postsynaptic cell membrane. Acetylcholine will bind to the acetylcholine receptor, or nicotinic receptor, on the postsynaptic cell membrane. As soon as this happens, the receptor will open its sodium-potassium ion channels and will allow positive ions to flood into the cell. If this ion flood is enough to bring the nearby voltage-gated sodium channels to threshold potential, it will cause these channels to open. The new action potential will be propagated along the postsynaptic cell, and at last we will have had a neuromuscular transmission. At last, our acetylcholine is taken by an enzyme called acetylcholine esterase, or ACE. This ACE rips apart our acetylcholine letter into choline and acetate. Choline is reuptaken back into the presynaptic cell, and acetate is washed away into the blood. Later, we will learn that another action will take place at the end plate of our muscle cell to cause a contraction. As always, thank you for taking the time to learn with us today, and remember to like and subscribe for more content.